Hello, welcome to Biology, the scientific study of life. More about the DNA. Evidence that viral DNA can program cells. Additional evidence for DNA as the genetic material came from studies of a virus that infects bacteria. Viruses are much simpler than cells. A virus is little more than DNA or sometimes RNA enclosed by a protective coat, which is often simply proteins. To reproduce, a virus must infect a cell and take over the cell's metabolic machinery. Viruses that infect bacteria are widely used as tools by researchers in molecular genetics. These viruses are called bacteriophages, meaning bacteria eaters, or just phages. As in figure, as we show here. In 1952, Alfred Hershey and Martha Chase performed experiments showing that DNA is the genetic material of a phage known as T2. This is one of many phages that infect Escherichia coli, E. coli, a bacterium that normally lives in the intestines of mammals. At that time, biologists already knew that T2, like many other viruses, was composed almost entirely of DNA and protein. They also knew that T2 or the T2 phage could quickly turn an E. coli cell into a T2 producing factory that released many copies when the cell ruptured. Somehow T2 could reprogram its host cell to produce viruses, but which viral component, protein or DNA, was responsible? Hershey and Chase answered this question by devising an experiment showing that only one of the two components of T2 actually enters the E. coli cell during infection. The preparation for their experiment, they used different radioactive isotopes to tag phage DNA and protein. First, they grow T2 with E. coli in the presence of radioactive sulfur. Because protein but not DNA contains sulfur, the radioactive atoms were incorporated only into the protein of the phage. Next, in a similar way, the DNA of a separate batch of T2 was labeled with atoms of radioactive phosphorus because nearly the phage protein enabled nearly all the phage's phosphorus is in its DNA. This procedure left the phage protein unlabeled. In the experiment, the protein labeled and DNA labeled batches of T2 were each allowed to infect separate samples of non-radioactive E. coli cells. Shortly after the onset of infection, the cultures were whirled in a blender to shake loose any parts of the phages that remained outside the bacterial cells. And mixtures were then spun in a centrifuge, forcing the bacterial cells to form a pellet at the bottom of the centrifuge tubes, but allowing free phages and parts of phages which are lighter to remain suspended in the liquid or supernatant. The scientists then measured the radioactivity in the pellet and in the supernatant. It's clear here, inquiry: is DNA or protein the genetic material of phage T2? Experiment. In their famous 1952 experiment, Alfred Hershey and Martha Chase used radioactive sulfur and phosphorus to raise the face of the protein and DNA, respectively, of T2 phages that infected bacterial cells. One step: mixed radioactive labeled phages with bacteria. The phages infected the bacterial cells. Notice that this one represents phage and this one pink one, radioactive protein, and the cell or the notice that bacteria cell. Also notice that page 1 phages were grown with radioactive sulfur S to the power 35, which was incorporated into phage protein bank. Notice that. Stage number 2, agitated in a blender to separate phages outside the bacteria from the bacterial cells. Notice that this one empty protein shell. Notice. And this one is phage DNA centrifuge. Stage 3 or third stage centrifuge, the mixture so that bacteria formed a pellet at the bottom of the test tube. Notice that. And fourth, them or measured the radioactivity in the pellet and the liquid. Notice the pellet bacterial cells and contents. Batches 2 phages were grown with radioactive phosphorus P to the power 32, which was incorporated into phage DNA blue, this one. And notice centrifuge and pellet and radioactive the phage DNA in pellet. The result, phage proteins remained outside the bacterial cells during infection, while phage DNA entered the cells. When cultured bacterial cells with radioactive phage DNA released new phages with some radioactive phosphorus. Conclusion, Hershey and Chase concluded that DNA, not protein, functions as the T2 phage's genetic material. Hershey and Chase found that when the bacteria had been infected with the T2 phage containing radioactive labeled proteins, most of the radioactivity was found in the supernatant, which contained phage particles, but not bacteria. This host cells, or this result suggested that the protein of the phage did not enter the host cells, but when the bacteria had been infected with the T2 phage containing radioactively labeled DNA, most of the radioactivity was found in the pellet, which contained the host bacteria. This result suggested that the phage DNA entered the host cells, moreover, when these bacteria were returned to a culture medium. The infection ran its course, and the E. coli released phages that contained some radioactive phosphorus. Hershey and Chase concluded that the DNA of the virus is injected into the host cell during infection, leaving the protein outside. The injected DNA provides genetic information that makes the cells produce new viral DNA and proteins, which assemble or which makes the cells produce new viral DNA and proteins, which assemble into new viruses. Thus, the Hershey-Chase experiment provided powerful evidence that nucleic acids rather than proteins are the hereditary material, at least for viruses. Additional evidence that DNA is the genetic material. Further evidence that DNA is the genetic material came from the laboratory of biochemist Erwin Chargaff. It was already known that DNA is a polymer of nucleotides, each consisting of 3 components: a nitrogenous, nitrogen-containing base, a pentose sugar called deoxyribose, and a phosphate group. The base can be adenine A, and thymine T, and guanine G, or cytosine C. Chargaff analyzed the base composition of DNA from a number of different organisms in 1946 or 47. He reported that DNA composition varies from one species to another. For example, 30.3% of human DNA nucleotides have the base the base A, whereas DNA from the bacterium E. coli has only 26.0% A. This evidence of molecular diversity among species, which has or had been presumed absent from DNA, made DNA a more credible candidate for the genetic material. Chargaff also found apicular regularity in the ratios of nucleotide bases within a single species in the DNA of each species he studied. The number of adenines approximately equaled the number of thymines, and the number of guanines approximately equaled that number of cytosines in humans DNA. For example, the four bases are present in these percentages: A equal 30.3% and T equal 30.3% and G equal 19.5% and C equal 19.9%. The equivalences for any given species between the number of A and T bases and the number of G and C bases became known as Chargaff's rules. The basis of these rules remained unexplained until the discovery of the double helix. Additional circumstantial evidence was consistent with DNA being the genetic material in eukaryotes prior to mitosis eukaryotic, so exactly double its DNA content. And during mitosis, this DNA is distributed equally to the two daughter cells. Also in a given species, a diploid set of chromosomes has twice as much DNA as the haploid set. Okay, thanks for